Today's notes are on Punnett Squares. When we talk about Punnett Squares, we are looking at heredity, which is the passing on of characteristics or traits from parents to offspring. So basically, how traits are inherited, and this is usually occurring through sexual reproduction. The study of heredity is known as genetics. When we talk about heredity, a lot of what we know today comes from Mendel's experiments. Mendel was a monk who ended up taking really accurate notes of his gardening and how when he bred different plants together, he would get certain traits depending on those of the parents. And some of the things he learned were about dominant and recessive traits. So a dominant trait is the observed trait, which means that if the offspring has one of these alleles, then that is the trait we will see. And dominant traits are represented with a capital letter. A recessive trait is a trait that is covered by the dominant trait. It is represented with a lowercase letter. So a recessive trait, if you look here in this Punnett square, even though this parent has a recessive allele, that's going to be covered by the dominant and they'll still be purple. And then we also talk about genotype and phenotype. Genotype is a gene combination, so it is the letters that represent that organism's genes. And phenotype is the physical appearance, so it could be like purple, short, tall, anything like that. So Mendel, like I said, studied pea plants. In this cross, both parents were true breeding, which means that they were both homozygous. And remember, homo means same. Another term for homozygous is purebred. So we will either have two dominant alleles, or two capital letters, or two recessive alleles, which are two lowercase letters. And those parents are going to either show the dominant or the recessive trait. So when we say true breeding, we have one parent that was homozygous dominant and one parent that was homozygous recessive. When we cross them together, we get all the offspring are heterozygous because they have capital T, lowercase t. And our phenotype, our physical appearance, would be tall plants because that is the dominant trait. Okay, and then when we crossed two heterozygous together, we get a variety of traits. So we have a quarter of the plants that are homozygous dominant, half of the plants are heterozygous, and a quarter of the plants are homozygous recessive. And then another term for heterozygous could be hybrid, when we have two different alleles, so one capital and one lowercase. Punnett squares get their name from Reginald Punnett. He designed the Punnett square, which is a system for estimating the offspring of two parents. So it's how we look at the possibilities for different traits that an organism might have. Monohybrid cross, remember that mono means one. So when we're doing a monohybrid cross, we are looking at the possible outcomes for a single trait. So we would take our two parent organisms and their genes, and we could, one at a time for each gene, predict what characteristics the offspring would have. When you complete a monohybrid cross, we start by determining the parent genotype, so there'll be a description in the question normally. They might say homozygous dominant, or they might just give you the genotype. We separate the alleles, which would happen during gamete formation. So you're going to split those two letters apart, and then we set them up on the top and side of the Punnett square. So one parent will go on the left side, and one parent will go on the top. And it does not matter which parent goes on which side, as long as you're keeping their pair of alleles on the same side. So I wouldn't want to put little t for one parent and capital T for one parent on the left. They need to be in their appropriate pair. So one parent here, one parent there, or vice versa. Then we basically drag and drop the alleles together to predict the possible offspring. So I would slide this T over, slide this T down, and then again, this would fill in this column, and this would go over. And you'll notice that we always want to put the capital letter first in that square because that helps prevent us from making mistakes when we're determining the phenotype. So then I would say my possible genotypes would be capital T, lowercase t, and that would be a 1-to-1 ratio because we have an equal amount of each. And my phenotypes, depending on the trait we're looking at, so let's say this is height, like in Mendel's experiment, all 100% of the organisms are going to be tall because they all have this capital T dominant allele. So let's think. What is the genotype of a short plant? It would have to be homozygous recessive, so 2 lowercase letters. And then why is this the only possible genotype for a short plant? It's because short is a recessive trait. So if we had a dominant allele, if we added a capital letter, it would cover up that short trait and give us a tall plant. Therefore, in order to show the recessive trait, we have to have all recessive alleles, or all lowercase letters. An important thing to remember with Punnett squares is that we are only predicting the possible outcomes. It shows us the probability or the likelihood of a certain outcome occurring. It's not a guarantee. So if we did a Punnett square, we might see that in this case, if they're both heterozygous, and this stands for father and mother, the F and M, this says 3 out of 4 children are offspring are going to have yellow peas. But that doesn't mean that if that plant had 4 pods, we would for sure get 75% yellow, 25% green. Just like a couple might have 2 children. And based on probability, they should have 1 girl and 1 boy. But we also often see families that either have 2 daughters or 2 sons. So probability is just the chance that something might happen. It's not a guarantee. So make sure you write your summary, include the key terms in your packet and 1 question, and then come get a stamp.